This is the second video on gene expression. So the last thing we talked about are the modifications made to mRNA. So reviewing, you're taking a DNA molecule, converting it into an RNA molecule. That is known as transcription. Then you are taking said RNA molecule and you are converting it into a protein. And that process is known as translation. Once RNA has been completed, once transcription has been completed, you made an RNA molecule. This RNA molecule needs to go through some modifications, the adding of that cap and the tail, and the removing of the introns and pushing those exons together to have your finalized mRNA molecule, which will then exit the nucleus, find the ribosome, and start to be read to become a protein. So that finalized mRNA molecule after your modifications will then start the translation process. Before we start talking about translation, I do want to discuss the 3 types of RNA that are going to be key in translation. We've already looked at one of these RNA molecules, your mRNA. This is the messenger RNA that you made during transcription and then had some modifications made. This mRNA molecule, once it exits the nucleus, will then attach to a ribosome where it will be read for the translation process. The second key RNA molecule is called rRNA, and this is your ribosomal RNA. It's called R for ribosomal, M for messenger, right? rRNA molecule, ribosomal RNA, along with proteins, is actually what makes up a ribosome. So a ribosome is made out of 2 things, rRNA and proteins. The third type of RNA that's going to be key is something called tRNA. The T stands for transfer. And the transfer RNA is the guy who actually brings in the amino acids that will start to make that protein molecule. We're going to see how this happens. So let's look at the process of translation. Again, this is going from DNA to RNA, transcription, and now we're taking that RNA molecule and converting it into a protein, translation. It takes place in a ribosome that is attached in the cytoplasm. It's going to occur in 3 steps, similar to transcription. You are going to have initiation, elongation, and termination. But each process is going to be a little bit different. So in initiation, you're going to have the 2 subunits of a ribosome assemble on the mRNA. That may sound a little weird. This is your ribosome here, but the ribosome is actually in 2 pieces. It's kind of like a stapler that is going to read and add amino acids as it reads the mRNA molecule. But it's actually in the cytoplasm, it doesn't look like this in the cytoplasm before it has an mRNA molecule attached to it. It's actually going to look like this, and then this guy is a separate piece. When the mRNA molecule comes floating about, these 2 pieces will come together, and then you end up having this with the mRNA molecule within it. Once the mRNA molecule has both ribosomes pieces attached to it, forming one mRNA molecule with a ribosome, then that tRNA that we talked about, that transfer RNA, is going to start by binding to the start codon, a start codon. So a codon is every 3 nucleotides. If you remember, an mRNA molecule can be long. It's not going to be as long as a DNA molecule. For starters, you're only read off of a gene, and then you took out a bunch of introns. But whatever that molecule is, let's say it's A, G, C, C, U, U, A, say this is your RNA molecule, it is going to read every 3 base pairs. And that is known as codon. So this will be 1 codon. Every 3 base pairs is a codon. you can see that here. And each codon will match to one of 20 amino acids. We're going to look at the genetic code in a little bit, but for now, understand that your mRNA is going to be read every 3 molecules, starting with the start codon. So your mRNA molecule is pretty long, and what your ribosome is going to do is it's going to have that mRNA molecule start to pass through it, and once it reads the first AUG, called start codon, tRNA will bind to it. This is known as initiation. Elongation will be the adding of amino acids for every 3 base pairs after AUG. So you start with AUG, find the first AUG, and then every 3 base pairs after that codes for 1 amino acid. Okay? It continues that way, coding for amino acids. That is your elongation. One end of the tRNA holds an amino acid to it. The other end of the tRNA molecule is going to be able to read the complementary bases. So the tRNA molecule that is holding the amino acid that corresponds to AAA will attach to it, to the ribosome, adding in that amino acid. Okay, that is known as an anticodon because it's going to hold the opposite letters that code for that amino acid. So elongation again is going to be the adding of amino acids as the tRNA molecules read within the ribosome that mRNA molecule, always starting after AUG, because translation, the elongation process, cannot occur until initiation, and initiation requires that the 3 base pairs AUG be read. Finally, that third step is going to be your termination. And in this case, termination stops. So the reading of the molecule, the adding of amino acids, stops as soon as you reach what is called a stop codon. So you start at AUG, which is your, you start at AUG, which is your start codon. Keep reading every 3 pairs. So every codon is every 3 base pairs until you reach a stop codon. Once you reach the stop codon, it doesn't matter if the mRNA molecule is still very long. Once it reaches that codon, it's going to stop reading. The ribosome will split apart and the mRNA molecule will just detach and float away. And then what you're left with is a polypeptide that was made. This polypeptide sequence is what you're left with. And this polypeptide sequence will then go through some transformations to become a finalized protein. Okay. Again, we have 2 great animations for showing this process in detail and to help you understand it. The first one is by Pearson, so it's found within your book and your additional things for your book and Blackboard. You can also hit on it here by clicking on the play button. The next animation is a fun one. Also, it's from the same people who made the transcription animation in real time, and this one is translation in real time. So it talks briefly about translation, and then it shows you how fast it actually occurs, how in a split second you can have the production of a polypeptide from the mRNA sequence. I highly suggest watching both these videos so you learn well the process of transcription and translation. There's also a good figure in your book, figure 14.24, that looks at transcription and translation in a review. So it summarizes it. Shows you here transcription starts, then RNA is processed. So once the DNA molecule has made its RNA molecule, that RNA molecule needs to go through modifications, the adding of the cap and the tail, and the removing of the introns. And then that exon made mRNA molecule will then exit the nucleus and start step 3, which is the initiation process of translation. It will then go through this translation, and then finally it'll detach, and you are left with a polypeptide molecule, a finalized polypeptide molecule. But that does not mean that you have a finalized protein. What you have left is a finalized polypeptide. Okay? And that finalized polypeptide will have to go through a few transformations before it can go any further. So at the end of translation, there is a polypeptide sequence. This is your polypeptide here. So you attached with the tRNAs, you attach these in a specific order, these amino acids in a specific order, reading the mRNA molecule to form your polypeptide. Your polypeptide then has to go through modification and folding to finally become a protein. If you remember from back when we looked at biological macromolecules, proteins go through several stages. Okay, the first one is called your primary structure, is your polypeptide sequence. So this here is your primary structure. This is what is made during translation, the primary structure of the protein, which is your polypeptide sequence. But as you already learned from proteins, when we looked at biological macromolecules, is that proteins will then go through a secondary structure, a tertiary structure, and in some cases a quaternary structure before you have your finalized protein. So it goes through foldings and twirlings and a lot of other stuff before you end up with your finalized protein. Now, a few things that I want to mention. So far, I keep talking about proteins are made on ribosomes, and the ribosomes are found in the cytoplasm, right? You have your nucleus, DNA makes RNA, and that is called transcription. The RNA molecule is then modified, and it will exit the nucleus through one of the pores, where it will have find a ribosome that will attach to it, and then a polypeptide sequence will be made, and this is known as translation. And I keep telling you that the RNA molecule goes into the cytoplasm where it finds a ribosome. But you all remember from back when we talked about the parts of the cell that rough endoplasmic reticulum has ribosomes attached to it, and that it is also important in protein synthesis. And that is true because proteins can be synthesized in one of two places. They can either be synthesized on a ribosome that is free-flowing in the cytoplasm, or it can be made on a ribosome that will attach to the rough endoplasmic reticulum. So if the protein that is being made, the mRNA molecule, remember during initiation, the ribosomes attach the mRNA molecule, and the mRNA molecule will let that ribosome know if that protein is going to be used within the cytoplasm or the nucleus, in which case it will be assembled, it will stay being made right there in the cytoplasm. But if that protein that is going to be made is actually going to be used within the endomembrane system or is going to exit the cell, then that ribosome would quickly move and dock on to or attach to the rough endoplasmic reticulum, and then the protein will continue to be made there. So think about insulin. You eat a sugary breakfast, and your body now has too much sugar entering the bloodstream. So your brain tells your pancreas to make a hormone called insulin, so that that hormone can go through your bloodstream and tell other cells to remove sugar from the bloodstream, thereby bringing your sugar level back to normal. So quickly, your pancreas needs to make a bunch of insulin. And so the way it's going to do that is by making a bunch of proteins that are going to be destined for exiting the cell. So again, this insulin, which is a type of hormone, but specifically a protein hormone, okay, it's a polypeptide hormone. it needs to be made in large quantity within that cell, but it's not going to stay in the cell because it needs to exit the cell. So the pancreas makes this insulin and is going to send it into the bloodstream. So those proteins that are being made, that insulin is going to be destined to exit the cell. Therefore, when the mRNA molecule flows out of the nucleus and into the cytoplasm, the ribosomes quickly come over and attach to it. But the ribosomes read, oh, this guy is going to make something that's leaving the cell. So it is going to attach to the rough endoplasmic reticulum. That ribosome will dock like a little ship, like a little boat. It's going to dock onto the rough endoplasmic reticulum. And then as that polypeptide sequence is made, it will actually grow into the rough endoplasmic reticulum. And then from there can move on through all the steps. Through the Golgi, get onto vesicle, go to the Golgi, get packaged and modified and whatnot, and then finally exit the cell. The way that proteins that are made to be attached to the rough endoplasmic reticulum, so if the protein is going to be exiting the cell, it needs to dock onto the rough endoplasmic reticulum, and it's going to use something called a signal recognition particle to do this. This signal recognition particle is going to escort the ribosome to a receptor on the membrane of the rough endoplasmic reticulum. It's kind of like a tugboat that is guiding that big ship to dock at a port. That's what your signal recognition particle does. It's a little tugboat that's going to move that ribosome and dock it on a receptor of the rough endoplasmic reticulum's membrane. And their translation will continue. So initiation, so that when that mRNA molecule attaches, the initiation starts, the entire ribosome will move over to the rough endoplasmic reticulum thanks to the signal recognition particle, and translation will continue while it's attached to the rough endoplasmic reticulum. And then this little polypeptide that's being made is going to grow inside the lumen, the interior portion of the rough endoplasmic reticulum. Here's a nifty little image showing you. So here we have a ribosome on this side here that is reading an mRNA molecule, and as it's reading it, it realizes that it is going to make a protein that is destined for exiting the cell. So it needs to dock on the rough endoplasmic reticulum. The signal recognition particle comes around the little tugboat. It attaches to the ribosome and brings the ribosome to a docking station, which is a membrane receptor on that membrane of the rough endoplasmic reticulum. The ribosome then docks to it, and that polypeptide sequence continues to grow. So the elongation process continues, but this time it's growing inside of the rough endoplasmic reticulum. And when the ribosome then detaches during the termination process and leaves that polypeptide sequence alone, that polypeptide sequence is already inside the lumen of the rough endoplasmic reticulum. Please review this in your book as well. This is actually a figure from your book, and so you can keep reviewing it in case you are confused as to how that process occurs. Right, the next thing we're going to talk about is the genetic code and mutations. I'm going to stop the video here so it doesn't get too long. And in part 3, we'll continue with the rest of the chapter.